Oui, on peut venir. Oui. Allez, so, hello everyone. Hello everyone. We are going to start. So, hello everyone in the room, both in the room and online. Thank you very much for being here. So we are here for the third edition of the Olivieri Conference. The conference has been intended as opening the series of seminaries in ecology and evolution in Montpellier, starting the academic year that just started a few weeks ago. The conference name pays tribute to Isabelle Olivieri, who was professor of population genetics here at the University of Montpellier, and she was also a researcher and scientist at ISEM. Her work advanced the field of ecology, genetics, and evolution quite substantially, both from a fundamental point of view, but also from an applied one, especially in the field of the biology of conservation of rare species. And coming back to today's conference, there are two final points I would like to make. First, I want to thank the Pôle AEB of the University of Montpellier, AEB for Agriculture, Environment, and Biodiversity, for the support in the logistics and the funding for this conference, as well as the series of seminars throughout the year. And finally, we are a new team, as you might have noticed, of volunteers organizing this series of seminars here, plus Stéphanie Bedom, who is unfortunately not able to be here today. I would like to thank the previous team for their time and dedication, and also thank you all in advance for your cooperation in order to continue making this series of seminars interesting and fruitful for everyone. So without further delay, I will let Mathieu introduce today's speaker, Ophelie Ronce. Thank you very much.
Thank you, Joseph. It's my great honor, pleasure to introduce Ophelie. Who better than Ophelie could actually be standing here for the Olivieri Conference. Ophelie was Isabelle's PhD student, working on metapopulation, dispersal, aging, extinction processes. So already starting to work on structured populations. And she also engaged in a lot of collaborations during this time. And she actually then went on to a postdoc to Austin to work with Mark Kirkpatrick, whom she met here in Montpellier when he was doing a sabbatical. Show that it's important to engage with the visitors when they come. So she did her postdoc more on the limits of adaptation and limits of the niche, so more into ecology. And then she was recruited to CNRS almost straight away. And so she arrived in Montpellier back in 2001. She's been here since at ISEM here. But she also went to a sabbatical to Vancouver. Right. So Ophelie works more generally on the limits of adaptation when populations are challenged by, for example, climate change or extinction. So her work is really about thinking that, making the links between ecology and population genetic processes. Ophelie's work is not only research itself, but it's also being active in societies and more generally collective endeavor. So she's been the president of the ESEB, the European Society for Evolutionary Biology, and vice president of the SSE, Society for the Study of Evolution, which is the American counterpart of ESEB. She's organized the joint congress, very memorable experience, I think, here in Montpellier with Yanis and Yanis. Is also supporting all sorts of collective instruments, like providing the FRB, Foundation for Biodiversity, Scientific, the Scientific Council of that, sorry. And generally, she's very active in everything collective here, both at the international, national, and also at the local level at ESEB. So thank you very much, Ophelie, for giving this conference, and the floor is yours.
Thank you, Mathieu. So it's a great honor, and it is also slightly stressing to talk in front of my extended working family here in Montpellier. So I've been a part of this community for 30 years, and it's really a great pleasure for me to start this series of seminars in this room where I attended many seminars for years, and so I would like to start especially by indeed I would like to say first thank you, not at the end of the talk, but at the beginning. Thank you to this Montpellier community, to which I belong for many years, and that I think this event want to celebrate by having something kind of a get-together event at the beginning. And so this is just a random collection of pictures of friends and colleagues attending a couscous party in my street across years. This community and this friendship and this trust with people here in Montpellier was really part of my happiness as a professional in this career, and I want to acknowledge that before talking about science, because it's also part of good science to have good friends and to feel included and loved and supported throughout your career. So to come to the science, today I would like to, so actually I apologize if somebody has already seen this talk, because I gave it at the joint conference in Montreal last year. And I thought that not many people from Montpellier were there, so I will do it again. And when I prepared this conference, I was thinking about different topics throughout my career that I could talk about. And one question that has been coming back again and again through different projects I've been working on throughout years is a question of evolutionary potential. And it's always something that is coming back. And I want to tell you about recent project about that, addressing two types of questions. The first one is, when is this evolutionary potential really limiting the persistence of species or population, and what kind of impact it has on the ecology or the demography of populations? And the second type of question I want to address during this talk is, what determines this evolutionary potential? What makes some species or population more prone to persist and evolve and adapt to a changing environment? And I will illustrate answers to this question or question around that with theoretical examples and modeling, because this is mostly what I do. But those models take inspiration from different plant species that are described, that are shown here, and I will tell you more about them later, that differ a lot depending on their life cycle. So the first species I will mention is Santora coramosa. It's a very rare endemic plant that is a cliff specialist. It's growing, it's found only in 6 populations in a very tiny range of less than 3 kilometers square near Narbonne on the Massif de la Clappe. It's long-lived, it's a perennial plant, but it flowers only once and it dies after flowering. So it's this type of life cycle is called monocarpic. The species is self-incompatible, has very poor dispersal capacity. As a result, the 6 populations are strongly genetically differentiated and exchange almost no gene flow, at least in the contemporary history of the species. So this species has been studied by people in the group of Isabelle and by many people around here, including this room, since 1994. And in particular, in our group, we have conducted demographic survey since then, and those demographic survey suggest that all 6 populations have some growth rate that is fluctuating between years, that is consistently below 1, so all populations are in decline, and this decline seems to be accelerated through years. So actually, as I mentioned, Isabelle Olivieri was the one who started this demographic surveys and more generally the study of this rare endemic species in the 90s. And our motivation to study that species was to showcase as well how evolution potentially impacted conservation biology. So what you wanted to do is to connect those questions about the conservation of species to more fundamental questions about the evolution of mating system, of dispersal, of metapopulation functioning, and the kind of fundamental questions she was interested in. And so this desire to merge conservation biology with evolutionary biology was very strong as a red thread in our research, and it's something that I think a lot of us inherited from in terms of research project. I think it's a research project that is still continuing and is still, to me, really relevant and interesting. And I wanted to mention this last publication that Isabelle co-authored with us just before passing away in 2016, where she defended this position of trying to integrate evolution with conservation biology. So since this survey started, as I showed you, the decline of the species actually accelerated, and something that has become very clear is that climate change is not making life easier for this rare plant species, which grows in very arid environment on rocks and crevices. And so especially in recent years, strong heat waves in summer is causing a lot of mortality of the rosette stage in the species, the population of Santoria. And this rosette stage is the most critical for the population dynamics of the species. And so colleagues in the team have modeled the extinction probability of the species in the near future, depending on different climatic scenarios. And what you can see here is that with increasing frequency of dry years, the probability of extinction in the short term is increasing a lot. And so there's about between 20 and 90% chance of extinction in 20 years. So there's an urgent need to act if the species is not to disappear quickly. And because of that, managers of the land where the plant is growing in the Parc de la Narbonnaise have asked the team to help them to conducting population reinforcement, just to buy time for the species to find solutions so that it will not go extinct immediately. And so those population reinforcement were conducted by collecting seeds in 5 of the 6 population of Santora, growing them in a common garden where they freely pollinated. So they were actual gene flow between plants coming from different populations, which does not happen in the field naturally. And then a lot of seeds were collected on those plants in this common garden and were reintroduced back in the natural population among the other plants in the population. So 40,000 seeds were put back in the natural populations between 2018 and 2022. Of those 4,000 seeds, only something like less than 100 remain alive today. But because the population is crashing so much, this number still represents about 10% of alive plants in the natural population. So it's some event of gene flow happening, not major, but still not completely insignificant. It's not clear that this minor addition of individuals in the population is really sufficient to change something demographically just by its demographic effect. But because what we reintroduced in the population were potentially product of between population crosses that generally were isolated for a number of generations, we could expect that there will be some higher diversity put back in those populations, or at least some genes, some alleles that were not present in those isolated populations. And so we might increase potentially genetic variation for adaptive traits. And the question we're asking is it sufficient to reverse the declining trend of the population and allow the population to evolve adaptation to the changing climate and escape extinction, a process called evolutionary rescue. So this concept of evolutionary rescue was, the level of genetic variation necessary to achieve rescue of the population was very well studied and discussed in this particular publication of Richard Kowitz and David Hull. And this is the inspiration for the talk, the title of my talk, because I think this notion of demographic and genetic constraints on evolution are really what I want to tell you about today. So Dick Kowitz was also passed away relatively recently, and he was a major actor of this integration between demography and population genetics. So I also want to acknowledge his contribution here. And the model, the simple model described in this publication, consider a population that is declining. The population growth rate depends on some underlying quantitative traits that have some genetic variance. And those traits can evolve in this environment, and as the trait evolve to get closer to some new optimal value, the population growth rate will increase. And so the population growth will increase, and if it reach again positive value, the population will stop declining and will start growing again. But if this evolution is slow, as if you have very low initial genetic variance for the trait, the population will decline for a very long time before growing back again, and it might reach a population size where that is below the viable population size, or where the risk of extinction is very high. So the heuristic behind this model is that there's some kind of threshold viable population size. And if you go below this threshold population size, you will probably go extinct. If you have a high genetic variance, you will start to grow again sooner. And so you will stay away from this dangerous level of population size throughout the course of adaptation. And you can define the critical genetic variance, which is just a level of genetic variance that is necessary for the population to stay above this critical level during the whole course of adaptation. So that's a simple model of this paper. And the question we ask is that if we want to think about critical level of genetic variance in some kind of operational way, most species, as Centaurea corymbos, that have a complex life cycle. Like for instance, in the Centaurea, you have seedlings stage, rosette stage, and then flowering plants. And the transition between those stages depend on different life history traits, such as seedling survival, rosette survival, flowering rate, or fecundity. And it's not completely clear immediately how you would measure or define a critical genetic variance given this complex life cycle. In particular, the story is even more complex because not only you've got those different stages, which have a different ecology and sensitivity to climate, but within each stage, you also have variation between individuals due to size. So for instance, a given seedling can grow to a rosette of different size depending on its growth rate. And the following year, this rosette can grow or shrink to different sizes. And after a number of years, naturally, it will reach some size that allow the rosette to flower, but it could also grow to a bigger size before flowering, and then the flowering plants can produce seedlings of different sizes. And all those individuals have different contribution to the population dynamics. So those vital rates that determine the population dynamics are depending on size and age and stage in the life cycle. And from the huge quantity of demographic data we accumulated through 30 years of survey in the species, we can adjust different models to describe indeed how seedling size distribution changes, how the number of capitula produced by a plant is increasing with size, how the survival probability or the probability of flower changes with size and age. And so because globally larger individuals have a higher fecundity, a higher survival probability, and a higher probability to flower, it's quite kind of intuitive to think that if in the changing climate plants could grow faster and reach higher size faster, they could have a huge potential benefit for population dynamics and the prospect of extinction of the population. So the question is, so the idea that if the population could adapt and grow faster to bigger size, it might escape extinction. So we want to understand or build models that combine evolution with this kind of complex demography that is size dependent. And so this size dependent demography was modeled using models of integral predictive models recently, and this is a field that has developed a lot in population demography and population dynamics. And there has been different attempts to integrate these population dynamic models with evolution. And there's been a lot of controversy about the way it is done, because often in those models, the trait that is evolving is size itself, which actually is a difficulty in conceptualizing the model. And there's been a number of critiques of those evolutionary IPM models, especially those who model size as the evolving trait, that have been discussed in a number of papers. So the idea is that rather than making size the evolving trait, what we want to let evolve is more the growth function that describe how individuals change in size through time and age. So in our model, the probability to reach some size at time t+1 depends on your size at time t and your age. And you've got some distribution. So given your size and age at time t, you can end up in some distribution of size for the following time step. And we can adjust to data a function that describes this distribution that depends on a number of parameters. So the idea is just that, and this was the work of Adele Ehrlichman during her PhD, which was defended last year. So the idea to introduce evolution in this model is to say that we now have a parameter that is modifying the expected size given your size at time t. What is the increment in size you will have at time t+1? And we make this trait here called Z genetically variable, and we so increase the variation for this trait, and we ask how the variation for this growth speed, if you want, is affecting the evolution and the demography of the population. So the question we ask is how much variation in this trait can rescue the population from extinction. So we start a population corresponding to a scenario similar to the population reinforcement. So we start with seedlings of Xanthorrhoea corambosa. So we have a cohort of seedlings, about 500 seedlings, for instance, and we follow the fate of this cohort of seedlings for 50 years. So if we do that with our model that combines evolution and demography, we can look at how the trait controlling the growth rate of the individual is changing through time in different age classes. So what you see here on those graphs is for different age classes and for different levels of initial variants that were introduced in the cohort of seedlings through the reinforcement, the evolution of the growth rate. And so quite intuitively, in all age classes through time, the growth rate is increasing. So you're selecting for faster and faster growing plants because there are benefits of growing faster and reproducing faster and producing more seeds as you are bigger. And also what is quite intuitive is that almost every age class, not all, but most of them, if you introduce more phenotypic variants or genetic variants in the initial cohort of seedlings, you have faster evolution of the trait, and so you reach faster growth rates sooner. However, what is less intuitive and was a bit surprising is that you do not have the same distribution of traits in the different age classes. And in particular, the young age classes evolve to grow very fast. But if you look at the older age classes, you see that they grow less fast than the young age classes. So they have a value of the trait that is much less. And so what we see through the process of evolution is that initially everybody is growing at the same speed, but through time, with evolution, the older age classes grow much slower than the younger age classes. So, and this is not due to built-in senescence or aging in our model, because this is not in our model. This is really due to the process of evolution that is affecting differently the different age classes. And this is due to the very peculiar life cycle of Xanthorrhoea. Because I remind you that Xanthorrhoea is a monocarpic perennial. So what happens is that if individuals grow fast, they reach a size that is sufficient to flower, and when they flower, they die. So the only individuals that remain in old age classes are those individuals that grew slow enough so that actually they did not flower before. And so this is why through time, what you see in these older age classes is a non-random sample of the genetic variation you had in the population, and you have only the slow grower in the older age classes. So it's the same kind of effect that is discussed in demography, often in terms of a selective effect, where in the older age classes, generally you expect the best individuals, but in this life cycle, you expect the worst individuals in the old age classes, because they did survive, but they grew very slowly, because if they had been growing fast, they would have flowered and they would have died. So this evolution of faster growth rate has also consequences on the population demography and the distribution of size in the population. So what you can see here is for different age classes, you have through a different time point through the course of evolution, the distribution of size of individuals, depending again on the level of initial phenotypic variance you introduce in the population. So for seedlings, you see that the distribution of size is not changing because it's not affected by the evolution of the growth rate. But starting at age 3, 5, 7, you see that as you introduce more genetic variants initially in the population and the growth rate evolves to be higher, you evolve a distribution of size with larger plants. So the population start to be larger in every class. However, what you see also is that, so this increase in size has benefits in terms of demography because larger plants survive better, flower earlier, and produce more seeds. So if you introduce like some level of variance, you have more individuals in every age class. But this is not necessarily true if you introduce too much genetic variance initially, where you do evolve a faster growth rate, but the number of individuals you have is declining. And this is because if you increase too much the variance for the growth rate, you introduce a lot of individuals that are growing faster, but also a lot of individuals that are growing slower. And so you have some kind of genetic load due to the variance of the growth rate. And this is actually compromising the global growth rate of the population and causing a decline in the number of individuals. So there's some optimal level of variance that you can introduce without having a cost on the demography. So you can see that quite more clearly if you look at the total number of individuals in the population. So if you start with a population that is not genetically diverse and you introduce this cohort as we did in the population of Santorea, you see that you have a decline in the number of individuals, and after 50 years, your cohort is just extinct. If you introduce not enough genetic variation for the individual growth rate, you see that it does not change much, and you still have extinction of the population. But if you increase a bit more the genetic variation you introduce initially in the cohort, then you have a rebound of population numbers. But you can see that this happens only after about 20 or 30 years of decline. So the moment where the population will rebound again is in quite a long time, and the extinction probability, I told you before, it's about the same kind of a time horizon. But if you introduce too much genetic variation, you actually accelerate the decline of the species. So this modeling exercise suggests that there's limited hope for the Santorea Corimbosa because we can indeed show that introducing more genetic variation can, for the growth rate of the individuals, can reverse the decline of the population. But this happens only for a narrow window of genetic variation. It does not happen before for at least 20 years, which might be too long to avoid extinction. And those simulations are overly optimistic because this was a kind of a toy model still, where we ignored the fluctuation of environmental conditions between years, which is depressing even more the population dynamics. We ignore the fact that climate is warming, is still continuing to warm, so you have to adapt to a changing environment. And we ignored all the early effects that are due to the reproductive system of the species. From a methodological point of view, this study is interesting because it shows a new approach to, I didn't provide any detail about that, to integrate evolution in integral projection models. That is quite efficient computationally, but needs to be checked against more sophisticated simulation to validate. So on the related subject, this first study showed that indeed the evolution in a structured population with different stages has a kind of a quantitative dynamics because of the interaction between the life cycle and the genetic variation. But we looked at evolution of a single life history trait, which was the individual growth rate. In the field, climate change is affecting many more different life history traits in the population demography. For instance, summer temperature is affecting the fecundity, the seedling survival, the rosette survival. But the change in fall precipitation is having a large effect on seedling survival and fecundity, and not so much on other life history traits. So this motivated us to ask questions about actually what kind of genetic variation is important for the rescue of a declining population. And this is potentially important because not only are different life history traits are affected differently by climate change, but also they make different contribution to population growth. So in demography, this is generally measured by a quantity that is called demographic elasticity, which measures the effect of a perturbation in some life history trait or transition in the life cycle on the asymptotic growth rate of the population. And so in the case of Centaurea, for instance, so this measures the contribution of a given life history trait on the population demography. The population growth rate is very sensitive to a variation in rosette survival in the Centaurea, and less so to change in fecundity and seedling survival, and even less so to change in flowering rate. So because those different traits have different contribution to population demography, we might expect that genetic variation for those different life history traits is having different effect on evolutionary rescue. And so this is a project that Julien Offresson is exploring during his PhD which is co-supervised with Olivier Coteau. And what he found is that indeed the genetic variation for different life history traits has different impacts on rescue. And so what this figure is showing here is the minimal population size the population will reach in the course of adaptation to an environment that is causing initially its decline as a function of the genetic variation for different life history traits. So for rosette survival, for fecundity, or for seedling survival. And here, this is just indicative, like some possible viable population threshold, where if you go below that, you think you have a high probability of extinction. So you see that you can, for rosette survival, which has a huge impact on demography, only a small amount of variation potentially is sufficient to increase and the minimal population size and stay above it for a long time. But also, if you introduce too much variation for this trait, you will again have a population that is never persisting. So you have a tiny range of possibility of genetic variants that allow rescue for this trait. And for those other traits, you need more variation, but you also have a larger range of variation that allows rescue. So the conclusion is that indeed, this notion of critical genetic variance depends on how genetic variation is distributed across different life history traits. So the follow-up of those theoretical projects is to ask, well, how much genetic variance did we introduce in the population reinforcement in Centaurea corambosa? And for that, we have conducted a large common garden experiment in the Terrain Expérimental, where we grew a lot of families, the same families from the same seed lots we use for the reinforcement in the garden, trying to measure genetic variation for different adaptive traits and life history traits to connect it to the models that we have developed. So one difficulty with this type of common garden is that it's hard to be sure that the quantity of genetic variation we measure in the garden is really relevant to understand limiting genetic variation in the field, because there's a huge plasticity in the life history trait and the morphology of the plant. So this is a Centaurea corambosa in the garden in Montpellier, and this is one in the field, and you can see that they really do not look like the same species even. But we'll see if we can extrapolate from this. Mm-hmm. So the second part of my talk is telling you about different questions, which were different models which were inspired by species with a very different life cycle. So Brassica rapa is an annual plant with a very broad distribution, differently from Centaurea corambosa. And there's been work conducted by a number of authors in North America using a resurrection ecology to grow plants collected from seeds at different times in the past to assess how the flowering time has changed in the context of a climate that is getting drier and drier. And so from those resurrection ecology experiment, authors found that plants from 97 flower about 3 days earlier than plants from 2014 when they are grown in a common garden and crossed with each other, which if you compare that to rate of evolution using paleontological data is relatively fast evolution. And so the question we ask is this fast evolution in response to climate change something that we should expect to be rare or something we expect to be common? And is the fact that we see those fast responses of evolution for traits such as flowering time something we could expect compared to evolution of any trait? So the reason why we were interested in the evolution of flowering time is that not only it's an adaptation to a different climate, but it also affects patterns of gene flow and mating patterns. So in particular, there's a strong assortative mating for flowering time in many populations. So for instance, if you look at those 2 trees who have opened their buds at very different times, it's quite unlikely that they will mate together because flowering time is very highly correlated to bud burst time in those species. And assortative mating describes the fact that you have preferential mating among individuals with similar phenotype. And so there have also been studies in the same plant community by Art Weiss showing that the correlation between mates for flowering time is, depending on species, between 5 and 6%, so it can be actually very strong. So this is a question that we addressed during the PhD of Claire Godineau, was co-supervised with Céline Deveau, and we asked whether assortative mating would affect the evolution of phenology in response to climate change. So there's been a number of models already starting in the 90s as well, trying to understand what kind of challenge populations that are under a continuously changing climate face. And the way those models work is that you imagine that have some trait with some optimal value, maximizing the population growth rate. And because of a warming climate, for instance, the optimal value of this trait is increasing through time over time. And what will happen is that initially you will have a well-adapted population where the mean phenotype is matching the optimal phenotype. And as the optimal phenotype is changing, this population initially is not responding much to a changing environment, and it starts to lag behind the optimal phenotype. And as this lag increases, the strength of directional selection on this population is increasing. And at some point, the population is evolving as fast as the climate is changing and tracking this optimal phenotype with a constant lag. And if this lag is too large, so as this lag increases, the population growth rate is declining. And if this equilibrium lag is too large, the population will not grow and will decline to extinction. So there's, again, the notion of critical genetic variance, because this lag will depend on the amount of genetic variation you will have in the population. And so there's a level of genetic variation that allows the population to keep growing, and a level of genetic variation below which the population cannot cope with the changing environment. So adapting is not enough to allow rescue in this context as well. So the lag, as I explained, is because genetic variation is allowing faster adaptation. The evolutionary lag will be smaller, genetic variation is larger. The lag will also be higher if the environment is changing faster. So those different curves correspond to scenarios where the environment is not changing, or changing slowly, or changing very fast. And so for a given level of genetic variation, what we found in this study is that the lag is the same with random mating and assortative mating. So if there was no effect of assortative mating on the evolution of genetic variance in the scenario that we studied, there was no effect of the mating system on the evolutionary lag. So again, as I explained, there's a critical lag where the population will start declining. And so for each scenario of environmental change, you can define a critical genetic variance. So the question really is, can assortative mating change the genetic variance in the changing environment and allow the lag to be smaller or larger than with random mating? So the prediction about the effect of assortative mating on the evolution of genetic variance are not trivial, because assortative mating is doing two things on a quantitative trait that is determined by many loci. The first thing that is quite intuitive and discussed in the literature is that because it favors mating between individuals that resemble each other, they also tend to resemble each other genetically. And so it creates a lot of genetic association within and between loci, which will inflate the genetic variance. So it creates positive linkage disequilibrium, and this will tend to increase genetic variation. But something that is not discussed much often is that assortative mating is doing something else, which is it's creating additional stabilizing selection by eliminating the individuals that are extreme in their phenotype because they do not find mate or because they do not mate with the most fit individuals. So there's stronger selection to resemble the most fit individuals in the population. And this additional sexual selection, in addition to natural selection, is actually depleting polymorphism at every loci. So assortative mating is making two things. It's inflating genetic variance by increasing genetic association, but it's decreasing the level of polymorphism we have at each locus. And the result of this tension is that if we look at what happens in a constant environment, so here you have the filled circles are what happens with assortative mating, and the empty circles are what happens with random mating. The level of genetic variation that evolves in a constant environment with assortative mating and random mating is almost similar. So those two effects compensate each other almost perfectly. And this was actually a result that was discussed by Landy already in the 70s. But what was not discussed so much is that this is true in a constant environment, but in our simulation and analytical model, when we make the environmental change faster and faster, then we start to have differences in the genetic variation that evolves under random mating and assortative mating. And the genetic variation we maintain with assortative mating is always much higher than the variance we maintain with random mating. And as a result of that, the lag under assortative mating is much less than with random mating. So we indeed find with historical studies that in a changing environment, assortative mating is maintaining more variation, and this allows fast evolution, which is not the case with random mating, and could explain why for many species we see fast response of flowering time to climate change, why we don't necessarily see such fast evolution for other traits that are involved in climate adaptation. So what is interesting is that, so in this first paper we published during the PhD of Claire, we had looked at one type of assortative mating, which is assortative mating by time, which means that plants that mate with each other are only plants that flower at the same time. There are different ways to model assortative mating. You could also imagine an animal population where you've got preference for individuals that resemble you, and you mate with the most similar individuals, but there's no this isolation by time of mating. And so if you model those different types of assortative mating, you find different results in terms of evolution of the genetic variance. So here I showed you, so the lines are the analytical prediction and the symbols are the result of simulation. So for a given level of polymorphism on average, this is the genetic variance also in the literature of ontogenetics. You can see here the total genetic variation. And so for random mating, those two quantities are not very different because there's very little linkage disequilibrium, slightly negative because of stabilizing selection. But with assortative mating, so assortative mating by time in red or associative preference in yellowish orange, you have much more, the genetic variance is higher than the expected based on polymorphism because of positive linkage equilibrium. But if you look at the simulation, because the polymorphism is less with associative mating, again, you have almost, if you compare the symbols, you have almost no difference in the total level of genetic variance, depending on the level depending on the mating system. And even sometimes you have less with associative mating than with random mating. If you're in a changing environment, you here have again the total level of genetic variance as a function of polymorphism for random mating and for associative mating by time and by preference. And what you see is that you see this higher level of genetic variation with associative mating only for associative mating by time, but not so much for associative preference. And even often it's less even in a changing environment, depending on the type of associative mating. So there's really something about associative mating by time, which is maintaining more variation. And I think it's, to me, it's linked to ideas of a soft and hard selection, because there's something protecting the polymorphism in the fact that you have mating pools that are separated in time. So to conclude about this part, we found that indeed associative mating for Floritime results in the evolution of larger genetic variance in a changing environment, but not in a constant environment, and not for every type of associative mating. And that this higher variance could reduce adaptive lag and increase fitness under climate change, but only for traits that are under associative mating by time. And it could explain why we see this frequent response of flowering time. Last project I wanted to mention in passing. So this was a project that was started with many actually master students. So Hugo Tessier worked on that, but Philippe is here as well. So this is a project asking how the stage structure in the population is also affecting the evolution of genetic variance for different traits. And this is just showing the amount of genetic variation and phenotypic variation and covariation of a trait that affects the flowering probability. And if you could measure these traits in seedlings, in rosette, and in flowering plants. So it's a trait that is expressed when you flower, but it's traits you can measure in all the stages. And so what is interesting in this scenario is that we find, so we assume that there's an optimal value that makes individuals flower earlier. And so what you see here is that you have more genetic variation in the rosette stage than in seedlings and than in flowering plants. And this is a bit weird, because normally throughout the life cycle, what we expect is that through selection episode, you're losing genetic variation. So if you look at older individuals, often you expect less variation than in younger individuals, because you had selection removing genetic variant. And here you've got a stage where you accumulate variation. And the reason why we see that is because, so the individuals in the rosette stage that have a phenotypic value close to the optimum, they will flower earlier, and they will leave the rosette stage and enter the flowering plant stage. So they leave this stage, and the only one remaining in the rosette stage are individuals that are too high or too low phenotypic value away from the optimum. And so through selection episodes, actually the rosette stage is getting richer in extreme individuals. And so the genetic variation is increasing in this stage because of the stabilizing selection on the transition to the next stage. So this is an interesting phenomenon that we are exploring during Julien's PhD to try to understand how the stage structure is affecting the evolution of genetic variation. So to conclude and come back to this idea of demographic and genetic constraints, again, many studies show that the genetic and phenotypic variation for different life traits is the fuel for adaptation to a changing environment. But understanding this evolutionary potential does not require only measuring genetic variation. It requires integrating really population demography with understanding of genetic variation. It's only when you combine the two that you really understand the constraints on adaptation and the real evolutionary potential in terms of persistence and demography. And also, what this suggests to me is that despite decades of theoretical work on the evolution of genetic variation and the evolution of genetic variation, we still have a lot to understand and still a lot that we don't understand about this critical component of adaptation. And where we need, I think, more integration between ecology and quantitative genetics to better understand how the variation evolves and what are its consequences. And I will take any questions.
Very much. So you're feel free to ask him.
Well, thanks again for these very nice model systems and models. I just had a question on the Santoria modeling. So, is there any effect of growth rate on? And is there any direct effect on of growth rate on survival? Because these are plants that live in a very stressful environment. So maybe the faster they grow, the more susceptible they are to actually dying from drought.
Yeah. So we did not, in the modeling we did, there was no trade-off between, there was no cost of, there was no built-in cost of that the plants that would grow faster will survive less. We did not build that in there because it was more like a little bit of theoretical exercise to understand if you could rescue the population even without those trade-offs. But in the data, what we have, I don't know if you noticed, but seem to be a signal that if you are too big, you have a lower survival, which is a bit weird, but it might be related to this trade-off. So it might be still in the model because we have a survival function that is declining with size at some point. So maybe this is a consequence of if you grow too fast, you do not survive. And when you adjust those models, you cannot. So one difficulty we have, despite the amount of data we have, in the data we analyzed, we had difficulty adjusting individual effects to the data to distinguish between the fact that some individuals might be bigger and survive less, not because size itself is costly, but because some underlying traits is affecting both of them. But we could not fit an individual effect in the demographic data we have, so we cannot really tease that apart.
Along the same lines, do you actually know what the driver is behind the population decline? Is it temperature? Is it water?
Yeah. So the work presented, they analyzed how the different life history traits were affected by climatic factors in the field throughout the 30 years of data, and they could say what is the impact on the demography because they have a demographic model that says how the variation in this. So they did that, and so it's difficult to have a simple answer because climate is affecting many life history traits, and some have a huge impact and some have a lower impact. But the thing, I think, what is really deleterious for the population at the moment is that when it's very, very hot in the summer, like what happened in like those recent years where we had big temperature heat waves, it kills the rosette. And when it kills the rosette, it's bad because a plant has been growing for years and years in this environment and it dies before reproducing. So its evolutionary and demographic future is nil. And so when you start killing rosettes, which are kind of a resilient stage, I think it's really bad for the population. But we have fluctuation, also in recruitment, in seedling survival and everything. So it's both drought and heat waves.
Maybe we have one question in time.
We can try if we can.
Yeah.
Or Susanna, can you hear us?
Susanna, can you hear us? She can't hear us. They can us.
Okay.
Luis?
Luis? Okay.
Yes, the second part about said that the assortment by time was stronger than selection. Assortment, right?
Preference, yeah, yeah.
Is it possible that the effect of by time is more reciprocal? And if you have something that's also reciprocal,
What do you mean reciprocal?
like affects both partners the same way, like they need to be synchronized. Whereas the preference is in the case that it's mutual preference, then you have also stronger effect.
I think if I remember the equation, both of them I think, well, to me, in both of them, it's there's a function that determines what is the probability that the male will mate with a female. So the kind of asymmetry in terms of selection of partner is, I think, is the same. But really, I think what matters is that in the assortment by time, you compete for a given flower or a female only with males that are flowering on the same day. Not competing with every male in the population. And I think that's what maintains more variation, because you've got like small competition pool. It's almost like a special structure thing. While if you have the mating function that are used mostly in models of assortative mating, you compete with everybody, and then it does not maintain the variation as much.
Okay, we can try online.
Here math doesn't work, so I will ask it. Does too much phenological variance cause ecological isolation or cross-pollination between plants of Centaria? Is this a reason for population decline in the conditions of very high phenological variance?
There's two different answers to that. So in the modeling, we did not look at evolution of phenology, contrary to the second part of what I presented for Centauria. It's true that if you have too much phenological variance, you can not mate with each other. In Centauria, it can be an issue. The mating can be an issue not about phenology in the sense of when in the year you flower, but in the sense of in which year you flower, because if you flower 1 year and there's nobody else flowering that year in the population, you will not have a self-compatible mate. So it can be an issue, but it's not in the model. It can be an issue for the population that individuals do not flower at the same time, but it's not necessarily within the season. It could be across years. Because, yeah. So the reason why variation was causing population decline in the simulation, in the model, just because but it's a well-known result that if you have stabilizing selection on the trait and you have a lot of variation, you have a lot of individuals that are very maladapted. So you increase the evolutionary potential, but you also depress the performance, the mean fitness of the population, because you've got all those extreme individuals that are really poor, actually. So all the simulation I showed might exaggerate that, because what will happen is that also this excess of variation will be depleted by selection through time. So you might have a huge initial load and then will decline, and you have less of a demographic load through time. And initially you will have this demographic load. No.
So, in your introduction on the second point, you mentioned the fact that the reintroduction did not work as expected. So, given your theoretical results that you show afterwards, would you like clearly conclude that this was because we introduced too much genetic variance, or was something else?
No, because so the survival of the seeds we reintroduced was kind of similar to the survival of seeds in natural populations. So it's just that there's a huge mortality at this stage. So even if you introduce 40,000 seeds, you will end up with a very few individuals in the end. It's just the normal demography of the species. Question is whether, and in animals, they have this kind of question when they do reintroduction, and sometimes they decide to introduce adults instead of juveniles because demographically it makes more sense to do that. But with plants, it's more complicated to introduce something else than seeds, especially in this environment where you cannot plant really the plant. But no, we don't know at all about the genetic variation that we put back. But for the moment, we were even hoping that actually the demography would be better because potentially because the population are really isolated, there are some indications that there might be some heterosis, like you have a higher fitness if you cross under some different population than within the same population. So we're also investigating that at the moment. But globally, at least initially, the first survival stage, it was not really different from what we have in natural population. It's just that it's really hard to put a lot of individuals because it's a lot of effort to introduce this number of seeds, and most of them will die anyway. So.
Yes. And I was wondering if there was any local adaptation. What's that system design?
Yeah, so there were experiment, previous common garden experiment where they looked at differentiation of traits. There was no strong evidence of local adaptation. Like they did, QST, FST comparison, for instance, and because the population are highly structured genetically, they have very high FST. The level of differentiation we see for quantitative traits is generally less than expected for neutral traits. So, and there was no reciprocal translocation or something like that. There's some differentiation for some quantitative trait, but it's unclear whether it's drift or stability selection, and there's no strong evidence of local adaptation at all.
Do you have a signature of in Centaurea of establishments of seedlings, which is just special years, or is it trickling out every year a little bit?
No, there are variation among years of.
Right.
Yeah.
But then don't you expect to have a big, strong selection for a given genotype to have plastic blooming time so that they cover several years with a given genotype? Pet hedging.
Yeah.
Audience, according to the year and repeated success, which means that to maximize the fitness of the genotype, it's good that spread out over time.
Yeah, so heteroparity in itself is a kind of
Yeah, but there it's not heteroparous.
It's not heteroparous. We don't know yet.
That's spreading.
Yeah, so we don't know. So we'll know from the quantitative genetic experiment how much genetic variation we have for the.
There is a complementary question.
Yeah.
Which is obviously, do all seeds germinate the first year, or do you have dormant seeds?
No, there's no dormancy. Almost no dormancy.
No dormancy.
No. But the plant flower at different ages. I don't know whether it's it would be hard to tell whether it's passaging or it's just that they grew in different conditions and they could grow fast or not, because the flowering probability really depends on size and age. So the strategy, like if you small but old, you start to flower anyway. But I don't know how much variation we have around that, and it will be hard to decide whether this variation itself is selected for, or it's just the micro-environmental effect or whatever.
Going back to the assorted mating.
Yeah.
So how is it that you do the contracts with random mating in this time dependent process? These cohorts are forming over time and sort of not falls from this. Because it's smooth that more Oil aspect.
Yeah. Yeah. So that's the beauty. Yeah. So the beauty of simulation is that you can
What is the climate changing condition that you had that would lead to increased sort of
so there was no increased associative mating. It was like what we
I pick it up by, I guess you said, the mating is increasing the genetic variance and consequently under these points.
no, so there's no so in those simulations there's no evolution of associative mating. We what we did is we just look at the impact of the mating system on the evolution of a trait that is flowering time, but this is not determining the intensity of associative mating, because what determines the intensity of associative mating is the width of the the duration of flowering, and this was not evolving in the simulation. So by controlling this width, we could make strong or weak associative mating, but we could also make random mating by artificially mating plants independently of their flowering time.
Done some modeling of flowering time and counting for environmental effects. From both of
Yeah.
and enhancing temperature, squeeze.
Yeah.
Everything flowers on more of a similar day or duration.
Yeah.
I think there's more balance in selection.
So
when you start compressing all that variation in a few days, it's supposed to just long duration. You're removing a lot of that.
So because well, in your scenario, what's happening is that, so what we say is that associative mating in itself, if flowering duration is not changing, and if there are no environmental effect on directly on the flowering distribution, it will help maintaining more variation for flowering time in a changing environment. What you're saying is that environmental change itself is going to change its phenotypic variance, and this indeed could completely annihilate the fact you could maintain genetic variation, but it's completely cryptic because everybody is flowing at the same time for environmental reasons. So indeed, this is not in our model, and it could cancel it completely. But you could also, I guess maybe I'm not sure if there is only one pattern of reduction in variance with warming, or you could also have sometime increasing variance because of runtime reasons. So we did not address that.
We have one question online.
Yes, I'll ask it again. What about assisted migration for to new environments for Centauri? Is that a conservation or an option for conservation?
So the reinforcement are an instance of assisted gene flow, like we are modifying gene flow between populations at the moment by introducing individuals that are coming from other populations. There's no discussion with managers of the land of putting Centaurea somewhere else. I think the thing that this species is protected because it's very rare, but it has also patrimonial value because it's found only in this mountain near Narbonne. And so you could protect it. Potentially it could grow. I'm pretty sure it could grow somewhere else. But somehow I think the interest of people is keeping it there because it's a unique part of the flora in this region. I'm not sure that there's much discussion about, because it has no big functional role, like not like forest trees or stuff like that, of saving it by putting it somewhere. At the moment, there's no much discussion about that.
I had a question as an ecologist. I was surprised by the huge differentiation you have in the phenotype in the field in itself versus in the wild. So I was just wondering, the question is whether in this garden you had a flowering substance.
Yeah.
Whether you have any idea field what prevents it from using long-lived habitats? Is it herbivory or?
No, it's a very, very poor competitor. It's really like in ecology, it's a stress tolerant species that really cannot cope with competition. And so in the field and at the CEF, we removed all competitors by using TARP. And so it had a lot of soil and a lot of resources, which it doesn't have in the cliff. And it had some competition because they grew so big that they finally, competed against each other. But at least initially they were protected from competition. And if you go back to the experimental garden, so there were lots of seeds produced by those plants in the garden. But today there's no Santora growing around the experimental garden because they can't grow in vegetation. So they're really bad. It's really like the CSR triangle. They're really specialists of stress tolerance. And I think a lot of the specialists of stress tolerance that live in very extreme environment, climate change is pushing them, at the limit of their niche, really. So.
Yes.
To touch upon it a little bit, I was wondering this story about the width of this flowering period.
Yeah.
Which direction you would evolve in that system, and presumably it complicates also the way you measure actually associating, I guess. So, probably have you? So, is it do they make that associatively for? How could you model that?
So I show you some data. So generally what people do to measure the strength of estimating is they measure the correlation in phenotype among mates. Just
many flowers and timing of
yeah, so you depend on the trait. You can look at if you look at the mean flowering time or the onset of flowering. So what people do, that's what they measure. They take one trait. It could be size, it could be color, it could be level of education, and you correlate phenotypically the value of the trait in the two mates. And you can look at the strength of correlation. So that's what I showed. And this can be large or small. And it's known from models that if you measure this correlation, it will depend both on the variance of the trait and the width for flowering time or the width of flowering. But so if you change the width, change the strength of estimates. But also, it's true that if you change the variance in flowering time, you're also changing a bit the correlation as well. Everybody is flowing at the.
Which direction do you think it evolves?
I don't know. We wondered many times about having this trait evolve, but the difficulty is that you need a trade-off. Because flowering time, you have an optimal flowering time, you're going to evolve to this optimum if you independently. So for the width, the question is, do you want to keep the same total number of flower and you just spread them during the season? But if you just increase the width by not changing something else, you will. There's no cost. I mean, there's no cost of flowering for a very long time. You need to have something. You need to decide about the trade-off. Could be a simple trade-off, like you have a limited number of flowers, so you can put them all the same day or spread them during the whole season. Could be one possibility. I think Céline did some models like that at some point, but what important thing is that in our models, associating is not evolving itself, not directly.
Questions? Well, thank you very much. Thank you a lot, Ophelie.